All right, okay. So hello dears. Ayan. So I really should think of another entrance, no? Siguro sa second sa mga itoglaing parang opening. Okay, siguro balik-balik na. Okay. Hello dears, welcome. All right. Anyway, again, hello dears, welcome to yet another pre-recorded lecture in your clinical bacteriology laboratory class. And for this part, no, this is second to the last na gyud na pre-recorded lecture in Bacte. I'm so emotional, Char. This is the last, second to the last rather, pre-recorded lecture sa ato ang Bacte class. And we're still continuing, and it's the last dipod sa ato ang topics on specimen collection, handling, and processing. This is the last topic. And for this lecture, we're going to talk about anaerobic culture. So we're going to talk about how do we, again, culture mga, how do we culture specimens if we want to recover anaerobic organisms, all right? So, di ba, in your intro to micro, hopefully if not remember, sa Bacte and all that, in yung lecture classes, no, you have already been introduced to the different types of organisms, bacterial organisms, in terms of their requirement for oxygen, di ba? So, we have the aerobic, mga facultative, something facultative, blah, blah, blah. We'll also run through those terms again after this introduction. But yeah, diba, generally you have the aerobic organisms and the anaerobic organisms, right? And most of the topics natong i-discuss, from the start until now, from specimen collection until mga biochem testing, we only focused more on aerobic organisms. Because number one, it's some of the most common, or it's the most common mga isolates natong makuha in routine clinical lab. That's why we are focusing more on aerobic organisms. But again, we also don't want to exclude the anaerobic organisms because some of them, if not, some of them or most of them also cause very serious and life-threatening infections. That's why ato po silang gina-focus. But sometimes in the routine clinical lab, usually, dili kayo siya ma-encounter. Okay? That's why ato rang run through rata sa iyang procedures, and also because iyang procedures quite taxing and quite expensive and quite demanding. All right? So muna siyang dili kayo siya ma-encounter sa routine clinical lab. But still, that does not justify or that does not give us a reason na dili nato i-discuss. All right? So again, this is anaerobic culture. So, ato sa tag-space, Char, kaya wala may oxygen dito. Alright? So, muna siya ang theme. Alright, okay. So again, some terminologies. Again, we'll start first with, of course, anaerobe. By the name itself, anaerobe. So, without, aerobe. So, something or an organism that does not replicate or that does not live well in the presence of oxygen. Or, it lives well or replicates well in the absence of oxygen. Basta ang point lang is di siya ganhag oxygen. Alright? Okay. Obligate or strict anaerobe. When you say strict anaerobe, kailangan yun na walay oxygen. Kaya if naa, bisag gamay ra na concentration, mamatay siya dayon, majutaps siya dayon. Alright? Okay. So killed almost immediately in the presence of oxygen. Aerotolerant anaerobe, by the name of aerotolerant, makatolerate ra siya ginagmay. Alright? So, can survive some oxygen exposure, but dili gyapon siya makapadayon sa metabolic processes because again, oxygen is still there. Pero makaya ra niya mo tolerate o gamay. Alright? So, dili siya pare sa obligate na gamay ra na oxygen, mamatay siya dayon. Ang aerotolerant, bahalag na gamay na oxygen, makalive ra siya, but still, dili siya kapadayon sa iyang daily life, daily processes, because again, na oxygen. Alright? Okay. Ayan. So, aerotolerant, mga mo tolerate dyan sa inyong mga ex. Ha? Char. Wag mag-tolerate. According to Ellen Adarna, you deserve what you tolerate. Pak! O, di ba? Yes naman. Alright? Okay. Next, you have endogenous. By the name of endogenous, it's found within the body, no? Inside the bodies of animals or humans. And exogenous, outside the bodies of humans or animals, usually sa soil, sa environment, or mga other sources. All right. Now, so why are some, before we start with that, why are, anaerobes not, why are anaerobes? Or like, why do some organisms become an anaerobe? Because again, di ba, your anaerobes man good, okay, anaerobes, they lack two enzymes, okay? They lack the enzyme superoxide dismutase and catalase. Wala sila catalase, alright? Now, your anaerobes may lack both or may lack only one. All right. Now, these two are important in the degradation of your oxygen radicals or oxygen na byproducts because sa normal bacteria no na naani na duha ka enzymes, your oxygen man good are pag mo gamit sila na ay mga toxic. Ayan, sino mga toxic dyan? Toxic byproducts. Okay? Now, these toxic byproducts, all right, if wala ni na enzyme, dili ma-degrade ang toxic byproducts from oxygen metabolism into much more tolerable or much safer na mga byproducts na pwede rang ma-eliminate sa bacteria. Alright? And muna siya purpose sa superoxide dismutase and catalase. The superoxide dismutase, one of the toxic byproducts man good sa oxygen metabolism is the oxide anion. Alright? Now, this superoxide dismutase converts oxide anion to hydrogen peroxide. And of course, kaning hydrogen peroxide is still quite toxic, okay? Pero mas toxic yun ang superoxide. This is superoxide anion, alright? So mas toxic yun na siya compared sa hydrogen peroxide. Pero still toxic yapon ang hydrogen peroxide. So hydrogen peroxide, in the presence of catalase, di ba? Recall in yung catalase test, it will now be dissolved into water and oxygen. Water, oxygen ba? Nalimot ko. But these final byproducts are much safer, di ba? They are not, lethal. They are not causing harm to the bacteria. Kay water na lang man siya oxygen, di ba? So, muna siya purpose sa imuhang duha ka enzymes. Again, superoxide dismutase and catalase. Now again, nga namang ang anaerobes, dili sila ganahag oxygen. Alright? It's not that they don't really like oxygen. It's just that they cannot tolerate or they are not able to degrade the toxic byproducts from oxygen metabolism into much safer compounds. Okay? Na pwede rin nilang ma-eliminate. Alright? So muna siya point nga nung some organisms become anaerobes because they either lack both the enzymes or only one of the enzymes. And one of the enzymes. Now, both of them are important, again, in the degradation of the toxic byproducts of your oxygen. Okay? Please take note kinsa itong duha ka enzymes, superoxide dismutase and catalase. Alright, sorry, excuse me. Ayan, okay. Ayan, so we now go to the next part. We now go to the different endogenous, so mga inside the body, na mga anaerobes from various anatomic sites within the body. So ayan, so summarize ni Sir Jemu the best, okay? So sa skin, of course, you have the Propionibacterium species and Peptostreptococci. For upper respiratory tract, Peptostreptococci, Actinomyces, Propionibacterium, blah, blah, blah. Oral, intestine, as you can see, the intestine consists of a lot of anaerobes and the oral cavity also, as well as the genitourinary tract, right? So just try to familiarize them, okay? Di jud necessary na mo i-memorize kay daghan kayo, alright? But as much as possible, you're familiar with the various normal flora, endogenous flora, anaerobes sa inyong various anatomic sites, okay? Alright. Also, so under normal circumstances, if the patient is like living well, they're healthy, these normal flora na anaerobes, they do not cause infection, of course, because again, they are normal flora. But when, the patient becomes immunocompromised or they are subjected to mga invasive procedures that may have introduced these normal microflora na anaerobes to different sites of the body outside of where they normally live, then dito sila pwedeng makakos na og disease, serious or even fatal infections. So, why is it important that we know kung kinsa itong mga normal flora or normal microbiota na anaerobes sa atong anatomic site? Because again, it will help us predict the particular organisms that are most likely to be involved in the infectious processes that arise or adjacent to those sites. So example, you have lungs, okay? Tapos, di ba, respiratory tract, daghang mga normal flora na anaerobes. Alright. Now, let's say na ay mga invasive procedures or na other underlying conditions that may have introduced those anaerobes to the lungs, okay, na nagkakos na nun siyang infection ng patient. So, makaingon na ka na, okay, it could be coming from the respiratory tract, diri sa oral cavity ba, or gikan ba siya sa intestine, or asa ba, basta, inana. So, at least you already know, you have the knowledge kung unsay possible asa gikan, okay, ang infection, or asa gikan ang kagaw. Ngano siya naabot dito na organ ba or tissue, inana. Alright, so, muna siya importance, ana. Okay, now what are some predisposing mga conditions or factors or risk factors na pwedeng maka-introduce sa patient to these anaerobic infections, okay? So first is human or animal bite wounds because again, di ba, oral cavity, no? Your oral cavity contains a lot of anaerobes. So if ever ba gani ka na magsipaak, ay, or napaakan ka og animals, which of course, they also have their own normal flora na anaerobes in their oral cavity. So of course, pwede siya ma-introduce sa inyong skin, okay? And katong normal flora sa inyong oral cavity na wala na normally dito na sa oral cavity, na-introduce siya into the skin na lain na habitat or environment. So of course, mukha siyang infection. Kaya dili man na niya yung balay in a way. Alright? Okay. So next, aspiration of oral contents into the lungs after vomiting. Still the same kay from the oral cavity, normal niyang puyanan na transfer siya to the lungs, which is a new environment. Of course, mag-cause ng infection. Tooth extraction, oral surgery, or traumatic puncture of the oral cavity. Still the same reason. GI tract surgery or traumatic puncture of the bowel, of course. So if naay punctures, of course, mugawas ang contents of the bowel, which includes your, of course, the anaerobic bacteria in your anaerobes. Genital tract surgery or traumatic puncture of the genital tract, and introduction of soil into a wound. Ayan, introduction of soil into a wound. Especially, this is very, telling or defining of mga C. tetani, Clostridium tetani, or mga tetanus infections. Ayan. Alright. Because again, galing sa soil na ay spores sa imuhang Clostridium tetani, alright, which now leads to tetanus. Okay, so as you can see, most of the predisposing conditions kung kanos aka magka anaerobic infections is really more on parang traumatic puncture, no? Like nadyo mo traumatic or invasive na nahitabo sa body, alright? Because again, under normal circumstances, if intact ra gyud katanan dira, these normal flora na anaerobes, bisag asa na anatomic site sila located, they are really not causing any disease. Alright? Okay. Alright. Ayan. Now, we now go sa indication. So, like example, na infection, how can you say na anaerobe na involved sa infection? Okay. So, first is infection in close proximity to a mucosal surface because again, your mucosal surface is teeming with a lot of anaerobes, the normal flora. So, kung na infection near to the mucosal surface, then you may have already an idea na possible na anaerobe na involved. Okay? All right. Presence of foul odor, yes, because again, some of your anaerobes, they release odor in their metabolism anaerobically. Like na yung mga organisms na release of sulfur, all right? Na yung mga organisms na release of other bad odors. So usually, if na presence of foul odor, like gikan sa abscess, kana mga nana, or kana mga deep wounds, all right, and napajoy, foul odor, then that could be indicative of an anaerobic organism involvement, all right? Okay. Presence of large quantity of gas, still the same, because again, your organisms, anaerobic organisms, they also release gas in their metabolism, all right? Okay. Presence of black color or brick red fluorescence, because you later will discover kinsa itong mga anaerobes na mo produce of fluorescence, like mo fluoresce sila, mohatag sila color. All right, and presence of sulfur granules, still the same, or yellow granules as byproducts of their metabolism. Some of your anaerobes, they release sulfur granules. So mga granules, okay? Later, you'll know kinsa na sila. All right, okay. So after that introduction, we'll now go to the different acceptable specimens for anaerobic bacteriology or for anaerobic culture, right? Kung kada site, unsa rin mga kanang acceptable na mga specimen. Okay, all right. So for CNS, of course, and the rest, kamantay mo na these are usually aspirates, okay? Or these are usually kana mga gikuha jud from the deep wounds, okay? Because again, these are the areas that your anaerobes are really happy or they are really living there. They are really like kanang favorable in. Because again, these are areas na deep, so layo siya from the surface. So walay oxygen na available. Gets? So kabantay mo na for all the anatomic source, the applicable or acceptable na mga specimen sites are usually those that are aspirated, all right? Biopsied ba? Or kanagikan sa CSF good fluid, or still again, all of them, they are coming from deep sources. So like kanadjong not really superficial, like dili sa skin, right? So dapat jud katong deep, all right? Because again, these are the areas where there is no oxygen, or if naamagani less rang oxygen, and hence the areas where your anaerobes love to live, right? Okay. Ayan. So mga deep, mga How deep can you go? Char. How deep is your love? Charot. Okay, all right. For pulmonary, still the same. Aspirate, urinary tract, female genital tract, still the same. Also, for urinary tract is cooldocentesis. Your cooldocentesis daw, guys, is parang extraction of fluid from the pouch of Douglas. Yan, pouch of Douglas. Pouch of Douglas daw is parang a pouch behind the vagina of females. All right, I'm not familiar with the female genital tract. Not my cup of tea. Joke lang. Alright, but yeah, it's a recto-uterine pouch, posterior to the vagina. So, likod sa vagina. Alright? Okay. Alright, ayan. Tapos, sa pa may mga other, loculated abscess, meaning na ay mga pouches. Okay, na ay mga loculi or pouches. Ang abscess niya, kaya na po siya ginagmay ng mga pouch. Yan na ba? So, very, very conducive for anaerobic organisms. Kay grabe na jud ka secluded or grabe na jud siya katago from an environment, sa outside environment na really contains a lot of oxygen. Okay, all right. Ayan. Okay, now go to your specimen collection. So those are the different anatomic mga acceptable specimens per anatomic site. We'll now go to the different specimen collection and transport. So how do we collect? Okay, so first is of course the aspirate. Again, the specimen of choice ni siya for anaerobic culture or anaerobic studies. Aspirate. Kuhaan mo siya using a syringe and mo aspirate ka. Alright? Okay, so for aspirates, again, needle and syringe. Okay? Tapos, expel air in the syringe because again, oxygen, we don't want that. Kaya if anaerobes atong kailangan i-isolate, of course, dapat as much as possible, the environment that they are in, again, are really devoid or wala gyoy oxygen. Devoid sinas. Joke lang ew. Alright, okay. Place an alcohol soaked gauze pad over the needle because again, para, disinfection purposes lang Japon. Okay, inject aspirate into an oxygen-free transport. Again, preferably containing a pre-reduced anaerobically sterilized transport medium. We'll know about that later. Okay, and upon receipt, vortex. When you say vortex, imo siyang i-homogenize or imo siyang i-concentrate. Okay, vortex, imo siyang i-spin. Alright, okay. And yeah, that's for aspirates. Okay, next is blood, of course. Blood must be cultured. Still the same sa atong blood culture na to ang diba? Topic, alright? Obtain blood sample, blood culture bottles, and transport immediately to lab and incubate. Alright. Next is for tissue and bone fragments. So example, mga tissues, kanang na mga, infections sa bone. Alright, so of course is you biopsy. Tapos what you do is you place in an anaerobic transport containing pras. Okay, do not tilt or tip the container. If large pieces, dapat in a sterile container with wet gauze. Transport in pouches with an oxygen-free atmosphere. Again, still the same reason, para mu-live rato ang anaerobic organisms. And upon receipt, place 1 ml in a thioglycolate medium. And, or pwede mo siyang ibutang o grinder. And add that piece of tissue. Or imo sa siyang idurog ba? Imo siyang pagamyon sa. Alright? Para mas easy siya i-process. Okay? Kaya dili kayo siya dagko na tissues. Especially if dagko na tissues, you homogenize or using a grinder. Ay, okay. Ibang grinder alam ko. Joke. Okay. Homogenize until suspension is thick. So, imo siyang i-homogenize, imo siyang durugon. Okay? Alright? Using a grinder ba or vortex or other instrument. Okay. Alright. And again, preferably dapat in an anaerobic chamber. Still the same para walay oxygen na ma-introduce. Okay, for swabs, of course, as mentioned, generally they are not recommended for anaerobic culture because again, your swabs are usually for superficial na use. And aside from that, your swabs also may contain materials that may be toxic to these organisms. So, di ba, katong cotton. So, as much as possible, if mag-swab ka, you use katong mga synthetic like Dacron, Rayon, di ba, Dax, charot, okay. Alright. Pero if di good possible ang biopsy, then pwede siyang gamiton. You obtain the swab specimen, pero dapat ang transport yun niya is under anaerobic conditions. So, dapat nasa pras ba siya, nabasa siya sa anaerobic chamber, na pag-transport, okay. And upon receipt, place the swab in a 0.5 ml thioglycolate broth and a vortex. So, ang vortex, again, imo siyang imo siyang parang ipa-spin gamay para again ma-concentrate itong mga kagaw na naasa swab. Alright? Okay. And press swab firmly against the wall of the tube to remove as much as fluid as possible. And again, as mentioned, dapat synthetic fibers ang gamiton, not cotton or other material. So dapat rayon, calcium alginate, Dacron, and ato ang shaft. Yes, ang atong gunitanan is dapat plastic. Diba? Recall, some organisms like chlamydia and other organisms also na anaerobes may be toxic maybe affected by wood, di ba? Katong wood na shafts or gunitanan. Alright? Okay. Ayan. And for transport systems, dapat again, your specimens should be transported as much as possible, as soon as possible rather, and dapat minimum exposure good to oxygen. And again, they must not be refrigerated because refrigeration reoxygenates this system. So, refrigeration muhatag nun siya oxygen. So, dapat dili siya refrigerate. It should be transported as soon as possible or immediately, and dapat under anaerobic conditions. Okay. So these are the different methods of anaerobiosis or anaerobic environment, no? Pag-transport. So pwedeng anaerobic transport for swabs, di ba? Kung kailangan dyan mag-swabs with pre-reduced gel. Anaerobic transport for liquid specimens, imo sa siyang ibutang og murag package or packet na murag plastic para dili siya ma-expose again to oxygen. Anaerobic transport vial if tissue, okay? And for pouch, pwede pong pouch for transport of specimens. Okay. All right. Now, we have what we call your PRAS media. Now, what is the meaning of PRAS? PRAS is, again, pre-reduced anaerobically sterilized media. Again, these are media that are packaged, manufactured, and shipped and stored under anaerobic conditions. So these are, dili siya katong normal nato na culture media, okay? These PRAS media are pre-reduced, meaning kukuhaan siya oxygen, alright? Tapos, gi-sterilize pa gid siya under anaerobic conditions. Okay, alright. So, prepared again by, pwedeng number 1, boiling, para again to remove oxygen. Okay? To remove dissolved oxygen. So, di ba? Example, na-mention ako sa intro to micro ninyo, ang inyong thioglycolate broth, di ba? Pwede siyang i-boil to remove dissolved oxygen. Number 2, pwede mo siyang i-autoclave to sterilize the mixture. And number 3, replace any air with an oxygen-free gas mixture. So, after all of that boiling, then sterilizing through autoclave pa rin. Tapos, instead of, para ma-oxygen-free yun siya, pwede siyang hatagan o gas mixture or meron siyang i-pump o gas mixture na contains other gases aside from oxygen. Basta dili na gyud oxygen. Okay? Alright, ayan. Now, for the next video, we'll now go into specimen processing. So, after specimen collection nato for anaerobic studies or culture, we'll now go to how do we process these specimens once they are received in the lab. Okay? Alright.